I'm Greg Albery. I'm a postdoc at Georgetown University. I'm going to be talking about this sort of nexus of factors. This is a series of photos of me in my past, at some point incidentally contacting an urban animal. I don't endorse having done this, but I kept these photos in basically to show that it happens surprisingly frequently, basically, that you end up sort of contacting animals in urban contexts in ways that could expose you to diseases. So this work was done with people from the Verena Consortium, which is a consortium of mostly early career researchers that I co-founded with Colin Carlson. Dan Becker is the last author, and you can read about this on bioRxiv if you want to. That's our Twitter as well, if anyone wants to see, wants to go and have a look at what we're doing. So there's a load of different reasons that urban environments might drive wildlife disease. Urban environments generally have more food, which often leads to greater wildlife density, which can drive greater disease. Often this food is actually worse quality, and there are lots of pollutants in the environment, which can weaken their immunity. It's also inherently closer proximity to humans. For obvious reasons, anthropogenic environments have much higher rates of contact with humans, which can drive greater exposure rate to these diseases. So all of these things should drive greater spillover rates, and over time we're sort of expecting that an urbanizing world is going to have greater disease as a result. But so far the broad analyses have been sort of slightly equivocal. We've found both positive and negative effects, and it seems like there might be some subtle things going on that we don't quite understand. And the stakes for this are actually really high because, as I said, it's rapidly urbanizing the world. People are increasingly contacting wildlife in these contexts, and this threat is likely to increase as time goes on. So therefore, it's important to understand a variety of things, including which components of spillover specifically are being affected by urbanization. How do our sampling processes succeed at identifying these processes? And do novel spillovers generally happen in urban environments, or do they happen in the wild and then get imported into urban environments? Both of those options are still possible for COVID. So the strategy that we chose to go and look at this was basically taking this broad pan-mammalian analysis of urban status in mammal species and trying to link each species' urban adaptation status with its parasite diversity to see whether more urban species actually tend to have more parasites, basically. We're answering these two questions. Firstly, do urban adapted species have more known parasites? And secondly, are more of those parasites known to be zoonotic because urban species basically spend more time around humans? There's sort of this intuitive expectation that if these species have been spending more time around humans, over time we will find out about more of their pathogens being zoonotic because they've spilled over into us. So the models I'll sort of breeze through, but if anyone wants to ask me more questions about them, you're welcome to. We used this aggregated data set that we published earlier this year, which is basically 4 broad publicly available data sets of host pathogen associations, which we sort of cleaned and harmonized taxonomically. And this is the distribution of the species that we have across the Earth, basically. So the response variables that we were using were pathogen diversity and zoonotic diversity specifically. And we had a whole suite of explanatory variables which were also derived from a load of publicly available data sets. So we had citation number as a measure of research effort. We had host order, phylogenetic distance from humans, sympatry with other mammals, body mass, a life history PCA to pick up whether fast-lived or slow-lived mammals have different pathogen diversities, domestication status, and urban adaptation status. So our hope was that basically by accounting for all of these different factors, we would be able to identify sort of conclusively whether urban adapted species tend to have more parasites. It was done on a binary scale where basically the Santini paper from 2019, they identified whether species are urban based on whether they can use urban resources, basically whether they either visit or dwell in urban environments, which is subtly different from what Rory and Dave and people did when they were basically looking at whether it was present in an urban environment or an anthropogenic environment. To go straight ahead into the results, what we did find is that urban mammals have surprisingly greater pathogen diversity, and that's true for the majority of subsets of these pathogens. So it's true for sort of viruses, helminths, bacteria, stuff like that. And the difference is really quite striking. And it's also true for zoonoses. And again, that's also true for a load of different subsets. So urban mammals do indeed seem to have more pathogens than non-urban mammals. There's a load of different possible explanations for this, but we're going to focus on these two different pathways, basically. So urban mammals might be in closer proximity to humans, which allows them to be more often sampled, which then leads us to have more knowledge about their pathogens. Or they might have weaker immunity or sort of different population dynamics, stuff like that, which does drive genuine changes in pathogen diversity. It's also worth noting that there's some sort of non-linearity and cyclic nature to this, where it's possible that because of species, certain species might have certain traits that make them more likely to be sampled. For example, bigger, more ostentatious species are likely to be more easily sampled by everyone. And this could mean that we're both more likely to identify their pathogens and more likely to know that they're urban species. It's possible that there are certain corners of the globe, corners of the phylogeny, where there are urban adapted species, but we don't actually know about it yet. What we actually do end up finding is, so this is the urban effect without including citation number. Citation number has a really, really strong positive effect on each species' known pathogens. And when you account for citation number, the urban effect really drops massively. It becomes sort of marginally insignificant. Well, it's still significant, but you could push it into non-significance if you tried, which we did. So the question really is how much of this is being driven by sampling effect at a species level and how much of it is happening at sort of broader macroecological levels. We threw a load of different models at this. We had zero-inflated models, multi-response models, we had spatial models, just basically trying to get under the understanding of this, this finding. This is the output from our spatial model, which basically found that there's these really strong geographic trends in pathogen diversity, with some surprising sort of low pathogen diversity in some areas where you wouldn't necessarily expect it, like Amazonia and Southeast Asia. What we suggest is that this represents a geographic sampling bias, basically. One of the reasons that this is important is because there's geographic sampling biases in everything. Notably, the Santini paper basically shows that there's these really strong sort of hotspots of known urban animals in Europe and in North America, places like that. And I think it's worth critically analysing whether we think that there are truly fewer urban adapted animals in sub-Saharan Africa, for example. I think this basically is a sort of geographic projection of sampling effort, and so the reporting of urban animals. One of the reasons that's important is because it's also perfectly aligned with the reporting for diseases. So this paper by Tofen at Eco Health Alliance showed these really strong hotspots of reporting in Europe and North America. And so it's possible that these processes are really strongly influencing our inference. So the sort of second component of our analysis was we wanted to find out whether more of their pathogens are zoonoses because they've been contacting humans. And we did this using a path analysis, which was basically several different linear models crunched together. And we're basically asking the question is, do they have a disproportionate number of zoonoses given their overall diversity? So there's one possibility, which is that urban animals have more pathogens, which just naturally leads them to having more zoonoses. Or their contact rates with humans might disproportionately filter that pathogen diversity so that more of them are zoonoses. And these are not mutually exclusive possibilities. What we actually end up finding is urban status obviously has this strong effect on pathogen diversity. Pathogen diversity, unsurprisingly, has a really strong positive effect on zoonotic diversity. But when you account for that effect, the urban status effect actually becomes non-significant and slightly negative. So what this is basically telling us is that zoonotic diversity is mainly defined by overall pathogen diversity, and it's quite hard to pick up the signal of human-wildlife contact in this context. So as sort of general takeaways, urban mammal species do have more zoonotic parasites, and this is largely due to greater proximity and greater study effort, correlated geographic biases, and probably not due to human-wildlife contact. We sort of are suggesting basically that there is this sort of universal underlying conflation between urbanization or anthropogenic activities, synanthropy, exposure risk, and sampling effort. And this sort of triumvirate is going to make it very difficult to actually conclusively conduct these pan-mammalian analyses that actually broadly answer the question about urbanization. As a result, we basically suggest that land use disease studies should consider correlated changes in surveillance as you're looking at changes in the land use. It's sort of quite intuitive, and I'm sure loads of people take it into account anyway, but as an area becomes more urbanized, becomes sort of more full of humans, basically, it's sort of natural that, therefore, it will become better surveilled over time. And particularly if you're investigating spatio-temporal changes in disease and changes in development, it should be really important to take into account this. It's not a static process, basically. So the question really is, how do we build large-scale generalizable analyses that are robust to this sampling bias? And the answer is, don't use this analysis, basically. This analysis was conducted at the species level and using parasite diversity rather than risk, which is not how spillover happens. It's not how transmission happens. It makes it much harder to account for biases that occur below the species level as well, things like geographic variation in prevalence, stuff like that. So what we actually really need is individual or population level infection data, like prevalence or burden, spread across urban, gradients, basically, and across much larger macroecological scales. It's basically a sort of slight shift in framework, going from the old model, which is sort of trying to fill in this host pathogen association map with more hosts, more pathogens, and then more known associations. What we want to do is we want to take this sort of set and we want to make it into this three-dimensional model where we have more and more data points basically for each host pathogen association so that we can test these things on that third axis. What's cool is that these data do exist. Like this paper that just came out was really, really cool by Cohen et al. They basically scraped these prevalence estimates from 7,000 wildlife populations, and they used sort of these literature searches and aggregated public data sets. And they revealed that climatic deviations predicted wildlife disease variation, which is not the focus of this talk, but it's very cool. And it basically demonstrates that these data are widely available. We can get them and we can build these data sets. We just need to start prioritizing it. And if we can end up making something like this, we should be able to test these associations much more easily, basically. And then we could build a future that looks like this, I think. Thanks very much.
Can people hear me? Yeah, great.